Welcome, Professor. Can you hear me?
Yes, I can hear you. Can you hear me?
Yes, welcome. Uh, Thank you for being here.
My pleasure.
So, I'm going to introduce you in Portuguese to the participants, right?
Yeah, sounds good.
One second. Então, pessoal, esse é o professor Brian Patrick Keany. Ele é professor assistente de psiquiatria na University of Rochester, com afiliações adicionais em neurociência, cérebro e ciência cognitiva. Ele usa psicofísica comportamental e neuroimagem funcional para investigar a percepção visual de objetos, tanto como ela funciona em pessoas saudáveis, quanto em pessoas com sintomas psicóticos. O doutorado dele é pela University of California, em psicologia, e ele tem doutorado em filosofia analítica pela Rutgers University, e um bacharelado em artes triplo, física e astronomia, espanhol e filosofia pela Universidade de Pittsburgh. Professor, you have 40 minutes, okay?
Sounds good. I guess I just need to share my screen. Oh, okay.
Yeah, we can see that.
So yes, I think you already heard my biography and my description of where I'm coming from. But we are a very inherently multidisciplinary group. And so I'm a vision scientist by training, but for the last 15 years, we've been focusing very much on schizophrenia and psychosis. Let me tell you a little bit more about schizophrenia, the burdens of schizophrenia. I imagine that most of this should be kind of obvious to many people in the audience, but it's a tremendously burdensome illness. So, you know, the prevalence of schizophrenia is not that high. It's less than 1%. It's probably closer to 0.5%. However, despite that fact, it is tremendously burdensome because it has an earlier onset and it packs people throughout their entire life. And so just to give you some statistics here, so there's a 7% suicide rate according to at least one study. Lifespan can be cut short by 25 years. 80% will be unemployed, 20% are homeless. They're 3 times more likely to be found in jail than in the hospital. And the recovery rate, it is possible to recover and to go into remission, but it's on the lower side. And also in terms of the financial impact, it's very high because of the fact that most of these folks are unemployed and in need of treatment and help from others. So despite this tremendous burden, we still don't know who will get the illness or why. And so there's an urgent need in psychiatry to develop a biomarker for schizophrenia. And so what is a biomarker? A biomarker is a characteristic that's measured as an independent or as an indicator of normal biologic processes, pathogenic processes, or responses to exposure or intervention, including therapeutic interventions. So it's just a very wordy way of saying that you want something objective about the person that tells you whether they have the illness, whether they are going to have a particular trajectory in that illness, and also how they're going to respond to particular types of treatments. So what kind of biomarker might be useful in this regard? So we give a somewhat unusual response here, and we say that actually dysfunction in the visual occipital system could provide a type of biomarker. So if you look at the brain in the visual system, in the occipital part of the brain, you can find markers of schizophrenia that can be very useful. That's our argument. So question arises, why vision? So, you know, there are many different ways you can look at schizophrenia. So why are we focusing on vision? There are actually a whole bunch of reasons to focus on vision. So one is just that we know a lot about how the visual system works. So, you know, PubMed is a source of literature on biomedicine. You know, huge number of items there on the PubMed database. You can go on it at pubmed.gov. And what you'll see is if you type these keywords in here, you will see that there's a lot more literature on vision than there is on, for example, olfaction or haptic or tactile perception, auditory perception. So we know a lot more about the visual sense than we do about any other sense. So what does that mean? That means that the computational neuromechanisms of vision are well characterized. It also means that the psychometric methods that we have for measuring vision are well developed. So it would seem like a natural place to look for dysfunction using the visual system. I should say that the search date for these entries was in 2021. However, I just did a search last night, and the trends are almost exactly the same. So this is a continuing kind of phenomenon where there's much more focus on vision. Another reason to focus on vision is just most of the cortex is cortex is devoted to vision. So this is a figure from David Van Essen. It's over 20 years old, but this is it still holds true. 20% of cortex is actually is V1. We're not even talking about V2, V4. We're talking about the earliest visual processing center in the cortex. 27% of the entire surface area of cortex is vision. So if schizophrenia is a brain-based illness, which most of us believe, then you should be able to find signatures of the illness in the visual cortex. So it's a very simple argument, but I think it's pretty compelling. Hopefully you do too. And then the other sensory areas obviously are much smaller compared to vision. All right, let's keep going. Also, vision is one of the most explored domains of cognition. So you can, again, go onto PubMed and you can plug these key terms in and see how many papers you come up with. And you can put attention, memory, language, and vision. Although vision is somewhat smaller than the other ones, it's still quite high. So you still have a lot of items on vision compared to other domains of cognition. But despite that fact, if you go into PubMed again, and instead of just adding those key terms, you can join each of those words with the word schizophrenia. So attention and schizophrenia, memory and schizophrenia, language and schizophrenia, vision and schizophrenia. So the intersection of those two things, you find way more papers and literature on attention and schizophrenia and so on than you do on vision and schizophrenia. In fact, you have 8 to 20 times more literature on these domains in schizophrenia than you do on vision. So that means that this has barely been studied, relatively speaking. And so there's a lot to unpack, a lot to understand, a lot to learn. So it's an understudied area. So we, in our group, we're trying to fill in this void. Another good reason to study vision in schizophrenia is that hallucinations are quite common. They're about 25 to 30% of people will experience hallucinations. So it's not as common as auditory hallucinations, obviously. Auditory hallucinations, about 60, maybe 2/3 patients will have those. However, non-hallucinatory disturbances are quite common. In fact, non-hallucinatory visual disturbances are as common as auditory hallucinations. So 2/3 of patients will experience perturbations or changes to their visual experience, including seeing false motion, seeing objects as having reduced contrast, visual snow, things like that, seeing faces as being disconfigured and so on. So that's another reason to think that the visual system is a useful way to understand this illness. So it looks like I did just kind of, I think I'm not getting my my animations here. I can continue with this, but there'll be a couple slides that might be problematic. So if it's possible to switch to the slides that have the animation, that would be ideal. But otherwise, I'll just continue. So In this talk, I'm kind of focused on two types of visually oriented biomarkers, and they're actually quite different, but they're also connected in that they have to do with the visual system. These are two very different portions of the talk. One has to do with functional connectivity, how the different parts of the brain talk to each other in sensory areas, in particular in visual areas. And I'll be going into that. There's a paper I published in the fall in molecular psychiatry, which talks about a new biomarker that's based in functional connectivity. I'll go more to that. That will be about 2/3 of the talk. And then visual shape completion, which is a behavioral task. That's very simple, but also we argue it still can be quite useful. And that will be about maybe 1/3 of the talk of the remaining time that I have. Okay, so let's talk about functional connectivity. So what is functional connectivity? Functional connectivity is developed through fMRI. So you put people in a scanner, they lie down, they keep their eyes open, and they do nothing, basically. That's why they call it resting state. And they kind of just relax. And as they're relaxing, you measure brain activity, and you capture brain activity at a certain sampling frequency. So typically, like these days, I would say every 800 milliseconds or 0.8 seconds, you get a new kind of measurement of brain activity. So the brain activity, for those of you who are less familiar, is blood flow. Blood flow, when you have increased neural activity in a particular part of the brain, then blood flow needs to go to that region to give the neurons energy so that they can fire more. And so fMRI indirectly measures brain activity by measuring blood flow. So blood flow is a proxy for neural activity. Now, you divide the brain up into a bunch of regions, voxels, or it doesn't have to be voxels, it can also be parcels, which I'll talk about. And so basically you measure the brain activity in that voxel over time. And so resting state scans can last anywhere from 5 minutes to 20 minutes or longer. And so you're sampling every 800 milliseconds, and you're getting for each region of the brain, and the regions are numbered there on the left, so 1, 2, 3, 4, and so on. For each region of the brain, you're getting kind of a fluctuation of activity, up and down, up and down. So that's called a time series. And that's kind of what you start out with, is you start out with a time series for each region of the brain. And then what you do is you do a giant regression analysis. And the purpose of the regression analysis is to kind of explain as much of the time series as you can, and then see what's left over. All right, so you put in, they're called nuisance regressors. And so head motion can change the values in the time series. So you put parameters having to do with head motion. Physiological noise, such as cerebral spinal fluid and white matter, also can enter into this. And also you regress out temporal trends. So maybe the scanner will cause a gradual increase over time in the time series and so on. So you're trying to explain as much from the time series as you can from known factors. And the idea is that once you regress those things out, what you have left over is the residuals. And the residuals of that regression corresponds to the spontaneous activity, the unexplained activity of the brain that is thought to be just the activity of the brain kind of just communicating with itself, and certain regions communicating with other regions. And so we determine to what extent activity in one region can be predictive of activity in another region. So if you have two regions, region one, that has a time series, and you get the residuals from the time series. Region two, you have residuals from the time series there. And you basically want to see if the residuals from the time series of region one can predict the residuals in the time series of region two. If it can predict, then that means that they're functionally connected. And if, for example, if they're correlated, just using a Pearson correlation, if they're positively correlated, then that means that there's some sort of positive information flow that's going on between those regions. And so you can do this between all the regions of the brain, and it gives you a functional connectome. And so you can literally tells you how any given region is connected to any other given region. So that's the idea behind resting state. Happy to answer more questions about that, but that's a basic idea. Okay. So the work that we've recently published has been inspired and motivated by some really influential work 10 years ago. And so there are 2 articles. One is cerebral cortex, one is neuroimage clinical, and these have been cited hundreds of times, if not thousands. There are 2 kind of main findings that these papers have shown, and I think very convincingly have shown. One finding is that the thalamus in the subcortex is more strongly connected to sensory networks in patients. And number 2, the cortex is more weakly connected to itself in patients in these sensory regions. So those are the 2 main findings. The effect sizes were medium to large. They were consistent across sites. They were consistent across the studies. And they have large samples and so on. So I thought it was pretty convincing. And so that's something that kind of we wanted to follow up on. Several questions arise from those earlier papers. One question is which sensory networks specifically are implicated? So the reason that question arises is because the functional connectome was derived through Pearson bivariate correlation. Problem with that approach for deriving the functional connectome is that it overestimates connectivity and it includes indirect connections. So ultimately, you want to know not just the direct connectivity, you want to know the direct connectivity, but you don't necessarily want to know if region A and region B are indirectly connected. And so those things are conflated by using Pearson bivariate correlation. So we're gonna kind of overcome that issue. The other is can these disconnectivity results be found similarly across different types of psychosis? For example, bipolar disorder with psychosis versus schizophrenia. Can you find similar results? And number 3 is can these results form the basis of a viable biomarker? Is there any way I can switch to the PowerPoint version? I did upload the PowerPoint version. The text is being included here in the PDF version. Is it possible to swap the 2 out real quick or no? Maybe not. Okay. In any case, there are certain things that we expect to see from a good biomarker. One is that the effect size should be large. It's pretty obvious, right? So you want a biomarker that clearly distinguishes your group of interest. It should be easy to interpret so you know exactly what it's saying. You know, a lot of machine learning methods and AI methods can be difficult to interpret. 3 is you want it to be robust to confounds. So this is kind of obvious. But the confounds issue is very, very important, right? So you don't want to be explainable in terms of medication. You want to rule out the effect of in-scanner motion, which is a well-known confound, and so on. So we'll talk about that. I can't read my text for PowerPoint 4. I'm trying to remember what that was, but you want it to also be able to predict, to be specific to a diagnostic group, and not just a general psychopathology. So for example, you want to have a clinical control group, basically. You want it to have good test reliability. So if it gives you one value at one time, gives you similar value another time. And you also want it to be able to predict patient status, not across sites, right? So if I build a model,
Professor,
yeah.
Sorry to interrupt you. We'll try to switch to PowerPoint.
That would be helpful.
Hi, Brian. Now you can
Hello.
see me.
Nice to see you. I'm well.
Well, nice to have you here. I will talk a bit about in Portuguese.
Yeah.
About the EEG measurements you talked.
fMRI,
O que tá
yeah.
falando? Okay, Brian, talking that you almost came here.
Yes, it's unfortunate I couldn't be there in person. I've heard many great things about Brazil, but I'm happy to be here at least remotely. So perfect. Now, am I able to click through it, or do you guys have to click through it?
Can you see it now?
Yeah.
Okay. Which page you want? Go. You tell when you want the next slide. Okay.
Okay. There's a live animation, so I'll be saying next. Okay. So so we'll do this slide. So as I was saying before, next. The obvious thing that you want your biomarker to do is to clearly distinguish your target group of interest. Next. You also want it to be easy to interpret, understand, and you want it to be biologically plausible. Next. You want it to be robust to confounds, which I was mentioning before. Next. You want to be recoverable from a brief scan session. So, being in a scanner is uncomfortable. You have to lie down, it's cold inside. You're constrained within a small space. Also, it's expensive. So you don't want to have people to be spending many, hours inside of a scanner. That's not what we want. Next. Also, you wanted to differentiate psychosis patients, not just compare to healthy controls, but also clinical controls to show it's not kind of a aspect of general psychopathology. Next. You wanted to predict the patient status across sites or studies. So if you build a model with a biomarker and predict patient status, ideally would be able to predict patient status across sites, using a different scanner at a different location, because that shows robustness. Next. And it can prove, you want it to show that it can prove upon other more standard methods of discriminating groups. Next. And you want to have good test-retest reliability, right? So you want to show that if it gives you a value at one point in time, it's going to give you a similar value at another point in time. Next. Okay, next. So questions that we address in this paper that we recently published, which I'll be discussing, is which sensory networks specifically are participating in these patterns of dysconnectivity. So thalamocortical hyperconnectivity and cortical hypoconnectivity. So those two patterns, which sensory networks are playing a role? We don't know which sensory networks are playing a role because the previous studies have used Pearson bivariate correlation to derive the functional connectome, which conflates direct and indirect connections. So we need to kind of improve upon those previous studies and use methods that remove the indirect connections, that focus on the direct connections. Next. The other question is, can this biomarker that we developed, can it be found across different types of psychoses, for example, bipolar disorder with psychosis or schizophrenia? Next. And can these results form the basis of a viable biomarker? So those are the questions we'll be addressing for the maybe next 20 minutes or so. Next. Next. Oh, sorry, that's a repeat slide. You can just go through the slide. It's a repeat. You can just go through this one. Next. Yep. Sorry, I don't know how we got a duplicate slide in there. Go ahead. Okay, so let's talk about the methods next. So in this study that we published, we adopted the Glasser Atlas. The Glasser Atlas divides the cortex into 360 regions. These are surface-based regions, and they are individuated on the basis of myelination, cortical thickness, and resting state connectivity. And this is published in Nature in 2016. And so we adopted this atlas for our study. And there are 180 different regions, cortical regions per hemisphere. Next. In addition, we adopted the brain network partition that was published in 2019. And what this partition does is it assigns each of these parcels or cortical regions to one of 12 different resting state networks. Next. Next. So each of these cortical regions is assigned to one of these 12 different cortical networks. Of these 12 networks, 4 of them are sensory in nature and will be focused on for this portion of the talk. Next. First, there's the visual 1 network, which includes primary visual cortex. They're shown there in blue with the arrows. Next. Next. Then there's a secondary visual network, which includes extrastriate areas like V4 and LO and the face area, shown in purple. Next. Next. Oh. And then there's the somatomotor cortex, which is shown in the sky blue, which is pre and post central gyrus, includes somatosensory cortex, motor cortex. Next. Next. And then there's the auditory cortex, which is in the temporal lobe, shown in hot pink. And so these are the 4 sensory networks that we'll be kind of focusing on for this portion of the talk. Next. In addition to the cortical parcels, each of these networks also assigns subcortical regions to these same 12 networks. So these are axial slices shown here in the middle. And so each subject is going to have 360 cortical regions and then 358 subcortical regions for a total of 718 regions. And again, each of these regions is going to be assigned to each of these 12 different resting state networks. Next. Next. And so the data set that we primarily rely upon for this investigation derived from the ACP early psychosis data set. This is a publicly available data set. You can download it if you want to. And again, ACP means Human Connectome Project. And so this publicly available data has 54 healthy controls, 81 non-affective psychosis patients, primarily schizophrenia, and 24 affective psychosis patients who primarily have bipolar. Next. And each of them underwent 4 resting state scans, and these scans were 5.5 minutes long. Next. It's pretty high spatial resolution, so it's a multiband pulse sequence. Each voxel is 2 millimeters cubic. Next. We did pre-processing with fMRI prep, which is publicly available. Next. And then we did nuisance regression. I kind of talked about this a little bit before, but you regress out from the time series all known factors so you can get your residuals. And so we regressed out motion and cerebral spinal fluid and white matter, for example, D-trend and D-mean that each run. We also removed the portions of the scan that were impacted by motion, which it can be a confounder. Next. Importantly, we derived the functional connectome through a graphical lasso method. And the graphical lasso basically is a way to dramatically reduce the number of indirect connections. It creates a much sparser network. And so it has lots of advantages as compared to Pearson correlation. And so by applying this graphical lasso method, you could generate a functional connectome for each person. So you get a 718 by 718 resting state matrix for each person. Next. And so this matrix tells you how each region, cortex or subcortex, will connect to any other region. Okay, so that's what it tells you. So once you get this matrix, then what you do is you want to calculate 2 values per network per person. So what are the 2 values? Next. First of all, you want to calculate cortical-cortical connectivity, which is the average connection weight between all cortical parcels of a network. Next. And then thalamocortical connectivity, which averages the connection weight between the thalamus and all cortical parcels of that network, right? So It's literally two values that you're getting per network, per person. And these values are generated just through simply averaging the connection rate. Next. So let's kind of show you more concretely how we get this. Next. So this is an example of a resting state connectivity matrix for a single person. This is just showing you cortical parcels. So this is a 360 by 360 matrix. The color codings on the outskirts of the matrix show you which networks the parcels belong to. Next, the triangle I'm showing you here corresponds to the connection weights that relate to the regions of the visual 2 network, extrastriate cortex. And if you average those regions in that rectangle together, that will give you the cortical connectivity value for that network for that person. So now you're seeing the somatomotor network, and that's a little triangle there, and so the regions within that triangle, if you average them, that gives you the cortical connectivity value for that person for that network. All right, so that's how we get that. So now I'm going to show you the results, which are comparing the groups on cortical connectivity. I'm going to show you the results for each of the 4 networks. All right, so these are inverse violin plots with overlapping scatter. And first I'm going to show you the primary visual network. Next. So next. So the primary visual network is shown here. And so the controls are shown in green, the non-affected patients are in red, the affected patients are in yellow, and the mean connectivity value is shown on the x-axis. And as you can see, in the x-axis, the groups are pretty much similar. I should say that each of the black dots on these graphs corresponds to a single person. Okay, just that's what the black dots mean. It's the cortical connectivity value for a single person of that subject group. And so what you can see is there's no differences, right? So the groups are very similar here. Next. The secondary visual network is a different story. So the secondary visual network, the controls have more cortical connectivity as compared to the other 2 subject groups. And this is significant. Next. And you get the same sort of pattern of cortical hypoconnectivity in the patient groups as compared to the controls. Next. But you get no differences in the auditory network. So these differences, this cortical hypoconnectivity in patients is primarily found in these 2 networks. And furthermore, there's no differences between the 2 patient groups on any network. So regardless of whether you have bipolar disorder with psychotic features or schizophrenia, either way, you're going to get these patterns of cortical hypoconnectivity. Next. Now we're going to look at the thalamocortical connectivity value. So again, this is looking at how the thalamus connects to each of these 4 sensory networks. Next. So again, I'm showing you inverse violin plots with overlapping scatter. Each dot corresponds to a single person. I'm showing you the 3 groups here on the y-axis. And for the primary visual network, we got no differences. Next. However, for the secondary visual network, we are getting differences. And now we're getting differences in the opposite direction, right? So patients are showing more connectivity between the thalamus and this network as compared to controls. Next. Also, the somatomotor network. Thalamus is more strongly connected to the somatomotor network in each patient group as compared to controls. Next. And then finally, the auditory network, no difference. So the group differences are primarily confined to these 2 networks. And now, again, to repeat, the thalamus is more strongly connected to these 2 sensory networks as compared to controls. And furthermore, the patient groups never differ from each other. Next. To show these results graphically, we collapsed across patient groups since the patient groups were not different from each other. And we these results are thresholded and averaged across networks, and these FD are corrected. And as you can see, the patients are showing hypoconnectivity, cortical hypoconnectivity, and the somatomotor network. Next, and the secondary visual network. And I should say here in this legend, the legend shows Hedges G, which is just the fancy version of Cohen's D. And you can see it's about medium to large effect sizes. Next, for thalamocortical connectivity, the thalamus, as you might expect from the previous plots I just showed you, the thalamus is more strongly connected. Next, to sorry, go back. The thalamus is more strongly connected to those same two networks, and the effect size is medium to large. And again, Hedges G is shown in the legend in the center, and so the colors correspond to how strong the effect size is. So there's more to say on this, but those are the main findings for now. Okay, next. So these results are interesting. They're significant statistically, but they're probably not good enough on their own to form a viable biomarker. So how do we make this better? How can we combine these results to form something that's a biomarker that can distinguish the groups more decisively? Next. 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 So what we did is we defined what we call a somatovisual biomarker. Next. And this biomarker is defined in a very simple way, which is what we call a difference of z-scores approach. Next. The way that you calculate this biomarker is as follows. For each subject, next, you average the thalamocortical values across the visual 2 and somatomotor networks, since those are the two networks that primarily were playing a role. Next. Then you average the cortical values across the same two networks. Next. And then you normalize each of those values across all subjects. The reason you normalize them is because they're on different scales. The thalamocortical value is about an order of magnitude smaller than the cortical values. So that's why you need to normalize them. Next. And then once you normalize each of those average values, next, you subtract them. So you have... go back. You subtract the two normalized values. So you got thalamocortical value, and so from that you subtract out your cortical value. Now, if patients have thalamocortical hyperconnectivity and cortical hypoconnectivity, then this value should be what? This value should be larger or smaller for patients? It should be larger for patients because they're going to have the thalamus is more strongly connected in patients to these cortical regions, and the cortical connectivity is weaker in patients. So this difference in z-scores approach should produce values that are larger for patients than they are for controls. Next. And sure enough, we get that effect. So this is Hedges G. You can see it's quite significant comparing the patients in red to the controls in green. And on the x-axis there, that's the biomarker value. So the higher up you are on the scale on the x-axis, the more patient-like you are in your biomarker value. And so really this is like the crux of this biomarker. It's just taking these two different values and subtracting them. Next. I would like to highlight the fact that this biomarker is extremely robust. Next. For example, if you are to exactly match the groups on in-scanner motion, the effect size actually gets larger, which means that the motion in the scanner probably weakens the effect. And so by controlling for motion, you can get larger effect sizes. Next. Also, this is very important. 22 patients in this sample were antipsychotic naive. They never had any medication. If you just look at them, the effect size also gets larger. And it's highly significant. Next. You can also look at patients who had no other psychiatric comorbidities, no history of concussion, no mild substance use issues, and no nicotine use. All right. So these are clean subjects, as clean as you can get. And if you run them, you also get a large effect size. Next. We also looked at a bunch of other potential confounds: age, sex, race, parental education, IQ, anxious and depressive symptoms near the time of the scan, stress levels near the time of the scan, pre-processing strategies. We tried different pre-processing strategies. Any way you slice and dice the data, you get effect sizes ranging between 1.0 and 1.2. Next. What about shorter scans? The scans that they underwent are 22 minutes in duration. That's not short. That's not a short time, right? So a biomarker would be more viable if you can show that the biomarker could be recovered in a short scan session. So next. So we looked at a single 5-minute scan, and we derived the biomarker from a single 5-minute scan. And once again, we got large effect sizes. So that means that you can scan them for a total of 30 minutes, including structural scans plus functional scans, and you can get this nice biomarker. Next. Next. Can it be replicated? So here's some additional questions. Whenever you are dealing with complicated data sets, people are always going to be skeptical. Oh, well, maybe you analyze it this way so you can get a larger effect. And can the results be replicated in other data sets? That's one question. Next. Is it specific to psychosis? Maybe it's just a general aspect of psychopathology, right? So people who just have any type of psychiatric issue, maybe they'll also show the same thing. This isn't specific to psychosis. That's the objection, potential objection. So let's next. Let's address these by looking at 2 publicly available data sets. Again, these are data sets you can download yourself if you wanted to analyze them. Next. Next. The first data set I uploaded personally from my own data set. It's on openneuro.org. Next. And it's 22 psychosis patients, 19 healthy controls. Next. And next. Use multiband pulse sequence. Next. ACP style preprocessing. Next. Which I won't get into. We use graphical lasso as we did before to derive the functional connectome for each person. Next. And we got pretty much the exact same thing. So this is completely different subjects, different site, different pulse sequence, different pre-processing methods, and we get almost exactly the same thing, which is encouraging. Let's see if we can get a third data set. Next. So then we analyze a third data set. This is a legacy data set that was collected in the mid-2000s, and we published on this data set in Science Advances in 2021. Next. And these data were collected 20 years ago. All right, maybe 18 years ago. And these are 35 ADHD patients, 36 schizophrenia patients, 93 healthy controls. ADHD patients are a clinical control group. So this is useful to look at, right, to see if it's a general psychopathology issue. Next. It's a single band legacy data set. Next. We use fMRI prep for pre-processing. Next. And we use principal components regression to derive the functional connectome, which I won't get into, but basically it's another method for deriving the functional connectome that's better than Pearson correlation and also does a better job of removing indirect connections. Next. And so here are the results. So the schizophrenia patients have higher biomarker values, not just compared to healthy controls in green, but also as compared to the ADHD clinical controls in yellow. The effect sizes are smaller, and we can talk about why that might be, but the most important thing is that these are statistically significant and in the right direction. Next. Next. So can it predict patient status across sites? So there are, in the ACP data set, the Human Connectome Project data set that we were talking about before, There are 4 data collection sites for that. Okay, one of them's in Indiana and 3 of them are in Boston. And the idea is you want to see if you could build a model for 3 of the testing sites, and then using the biomarker, and then predict patient status in the remaining data site. And you do that for all 4 sites. And you see how good it can do. Next. And then you can create, next. And it's called leave one set out cross-validation. And then you can plot an ROC curve, right? So if it's perfectly discriminating patients and controls, you end up with like almost like this sort of an L-shaped, L-shaped thing. Here, we're not getting that, obviously, but we are getting a pretty good group separation, the AUC value of 0.79. This is sensitivity and specificity in the low 0.7s. So obviously a lot of room for improvement here, but pretty good in terms of classifiers go for this population. So just so everyone's clear, sensitivity of 0.74 means 74% of the patients are identified as patients, and specificity means 70% of the controls are identified as controls. Next. Is it reliable? So how reliable is this measure, right? So if it's really noisy and it's giving you different values at different times, then it's not going to be very useful. fMRI is known to be a very noisy method, and so this is an important question. Next. So to get at this question, what we did is, we had these 4 resting state scans. So we calculated the biomarker from the first 2 resting state scans, and then we calculated biomarker for the second 2 resting state scans. They were separated in time by 35 minutes. Not that long, but still long enough. We wanted to see how consistent they were across these 2 in this interval. Next. And what we found was that intra-class correlation value was 0.62, which is moderately good. And for fMRI is very good because, like I said, fMRI is a very noisy method. So definitely room for improvement, but not bad. Okay, next. What is the cause? Okay, so we show these patterns. Thalamus is more strongly connecting to sensory areas, the somatomotor areas, visual areas. The cortex is more weakly connected. What's the cause? Why is this happening? Next. So we propose that the mechanism is that there's pathology happening at the NMDA receptor. And why would we say this? Well, one reason is that there's a ketamine study showing that when you give people ketamine, which is the antagonist to the NMDA receptor, the thalamus ends up hyperconnecting to parts of the somatomotor cortex. Next. And furthermore, it ends up hyperconnecting to parts of visual cortex. So these are the same regions that we're talking about. So this is very interesting. This is a pharmacological model of psychosis, and you can get similar results during a connectivity analysis. Next. This is further evidence that NMDA pathology, receptor pathology is responsible. So this is anti-NMDA receptor encephalitis. These people have an autoimmune condition that attacks the NMDA receptor, and it causes symptoms very similar to schizophrenia. And they did a resting state scan for these people after they were their symptoms started subsiding. And what they found is they found cortical hypoconnectivity in these patients. And the more severely affected the patients, the more hypoconnectivity that they found. This is the case for the visual cortex. Next. And it was also found it was also found for the somatomotor cortex. So the more affected they were by the NMDA receptor encephalitis, the more hypoconnectivity you would find. Next. So to summarize, next, regardless of diagnosis, psychosis patients had cortical hypoconnectivity. Next. Thalamocortical hyperconnectivity. Next. And this primarily impacted the visual 2 somatomotor networks. Next. And it could form a simple and robust somatovisual biomarker by using this difference of z-scores approach. Next. This biomarker can be recovered from a 5-minute scan. Next. It could be replicated into other studies. Next. It could predict patient status across testing sites. Next. It was moderately reliable. Next. And it's plausibly caused by NMDA receptor hypofunctioning. Next. Okay, now we're going to switch gears. So that was all kind of heavy neuroscience, right? Now we're going to talk about something that is a little bit more accessible, perhaps. This is just good old-fashioned behavioral psychophysics, right? Just behavior, having them press buttons based on what they see. But this sort of approach also can be useful. Next. So the various types of visual perceptual deficits that people with schizophrenia will exhibit. They'll exhibit deficits in eye tracking. They'll have more restricted eye tracking movements. Contrast sensitivity will be reduced, especially in the later illness stages. Depth inversion illusions are also reduced in people with schizophrenia if they're sufficiently psychotic. Backward masking is another type of phenomenon where if you see a target and then you see another kind of object that appears shortly thereafter, you're less good at seeing the target if you have schizophrenia. And there are numerous other types of visual deficits. So here I'm just going to focus on one type of visual deficit that I think is pretty interesting in schizophrenia. Next. 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 So the process of interest is visual shape completion. So these are called kinesis squares. The edges, next, are called inducers. Those inducers launch the formation of illusory contours, next, which are very subtle, but which your brain forms within 200 milliseconds, 150 milliseconds, next. And they form these illusory contours which contain the presence of illusory shape, next. So this is it's been studied since the 1950s, and it's very well documented. Next. So why, of all things, why look at visual completion? Well, it seems kind of random, right? Why look at visual completion schizophrenia? Next. One very good reason to look at it is actually it's really important for normal seeing, right? So this process, next, helps us recover object shape and number. So if I say, how many objects do you see here? What shape do these objects have? Oh, that's easy. I see 3 objects that are circles, right? Next. But now if I say, how many objects do you see? What shape do these objects have? You're not going to say, oh, now I just see 3 circles that turn into notched circles. You're not going to see that. You're going to say, I still see the same 3 circles, but now there's an overlapping white triangle that's on top of those 3 circles. So the shape and number of objects that we see and report is determined by this process of perceptual organization and illusory contour formation. Next. 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 This is a movie, so it might not show. Oh, you have to press, can you play the movie?
É filme.
If not, it's okay. There's a movie in the previous slide. You have to go put your cursor onto the movie and press play.
Conseguiu passar o filme? No.
No? Okay. But it also allows it so you can have these little tiny fragments that go in and out of existence. And your brain can take these little fragments and form these illusory contours to get a well-formed shape representation. So this is a super important and underappreciated aspect of vision that allows us to make sense of the world around us. Next. There are also reasons to think that perceptual organization is impaired in people with schizophrenia. So this is a first-person report. Next. 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 Go back. So this is a report from a paper from 1961. This is from a patient. A patient says everything is in bits. It's like a photograph that's been torn in bits and put together again. If you move, it's frightening. The picture you had in your head is still there, but it's broken, right? So there's some sort of perceptual organization process that is not operating as it should among certain patients with schizophrenia. So that suggests that this process of visual shape completion also could be potentially impacted. Next. Next. So the question that we address here is, are there robust shape completion deficits in schizophrenia? Next. And if so, are they diagnostically specific? Next. 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 So we had 3 subject groups. We had bipolar disorder patients, next. Schizophrenia patients, and we had, next. And we had well-matched controls, next. They are matched on age, sex, and parental education, next. And the task that we gave them was pretty straightforward. They discriminated these Pac-Man configurations, next. And there were 2 conditions. There's a illusory condition and a fragmenting condition, next. So for the illusory condition, you show them these Pac-Man. And so if you see here, you should see a illusory square, right? Next. And you rotate each of these Pac-Man by a little bit, either clockwise or counterclockwise. And if you rotate them in a certain way, they're going to form a fat shape or a thin shape. Next. That's a fat shape. Next. That's a thin shape. So the fat shape, thin shape. Go back, please. So fat shape, forward. Next. Thin shape. You can see the difference? Okay, so that's what the task was. And again, you varied the difficulty by how much they were rotated. Then we had a control condition, next, where the same thing is that the Pac-Man were pointing downwards to break the formation of illusory contours or shapes, next. And they were rotated all to the left or all to the right. And that was the task, to say left or right. Next. The trial sequence was as follows. You show a blank screen, next. Followed by the stimulus, next. Followed by a blank screen, next. Followed by a mask for 300 milliseconds, next. And that was the case for both the illusory condition and the fragmenting condition. In each case, you'd be shown a fat or thin shape or a leftward or rightward rotated Pac-Man, and you had to say which one that you saw. Okay, so fat, thin, or left or right, depending on which condition you were doing. Next. Next. So task difficulty dependent on the rotation. Next. So if their Pac-Man would just rotate a little bit, it was very hard to tell which one was fat or thin. Next. Whereas if they were rotated a lot, then it was very clear that one was fat, one was thin. Next. And threshold corresponds to the amount of rotation needed to achieve 80% accuracy. So we use an adaptive staircase method to determine how much rotation was needed to generate 80% accuracy. Next. Next. So these would be hypothetical psychometric curves. So that here's the threshold for the control. Next. Here's a threshold for the patient. Go back. And you can see that the patient has higher threshold, which means they need more rotational difference between fat and thin to generate 80% performance, right? So if you see these 2 curves, that means that your patients are doing worse. Next. The stimuli we used are as you can see here, we had fat and thin stimuli, and we also had left and right stimuli. We also had other conditions, which I won't be getting into because of time, in the interest of time. Next. But we also added distractor lines on certain trials. And again, I can discuss that if you want me to, but I'm not going to be focusing on that. Next. So here are the results. This is published in 2022 in schizophrenia research. Threshold is shown on the y-axis. So higher threshold means you're doing worse, right? So higher threshold means you're doing worse. Controls are in green, bipolar is in yellow, schizophrenia patients are in red. And I'm showing you the data for the lines and no lines. And what you want to see here is that the patients really have trouble on the illusory condition as compared to the controls with bipolar patients intermediate. Next. For the fragmenting condition, though, patients have very little trouble. They're a little bit worse than controls, but really not much. And you can see that all 3 subject groups are very similar on that task. Next. So next what we did was we defined shape completion as a threshold difference between 2 conditions. We took the threshold from the illusory condition, from that subtracted out the threshold from the fragmenting condition with no distractor lines. And that threshold difference corresponds to how much they're doing in terms of shape completion. Okay, so that threshold difference is what we're focusing on. Next. Here I'm showing you the fragmented minus the illusory. So you can do it either way, actually, illusory minus fragmented, fragmented minus illusory. For this graph, we're showing fragmented minus illusory. So if you're doing better on the illusory than you are on the fragmented, then you should have a positive value, right? So fragment should have a high threshold, illusory should have a lower threshold, and this should be a positive value. And you get a slightly positive value for patients, for controls, but you can see the patient group gets worse, right? So the patients really have a specific problem to shape completion as compared to the healthy controls. The bipolar patients, intermediate. Next. We also, just for robustness and we also just looked at the practice data. Could you find the same thing if you just look at the practice data where you show like a small number of trials at a set difficulty conditions? And we get pretty much the same sort of thing. The effect size is smaller because of smaller number of trials. We get the same thing using just, we're not using any staircase procedure for the practice. It's just method of constant stimuli. Next. And then we also looked at the group difference as it unfolded trial by trial. So you get a new threshold estimate after every trial using this adaptive staircase procedure. And you can see really you start getting maximum group difference around trial 30 to 35. So you'll only need 35 trials to find this relatively large group difference. Next. Another question. Okay, well, there are these interesting deficits. Do they arise by the early illness stages? Next. And do they continue to worsen after the first psychotic episode? Next. So this is another paper that we published in Journal of Normal Psychology. Next. Oh, and also, are they linked to illness features? Next. 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 So we had 23 first episode patients, 49 later episode patients, 48 back, 48 healthy controls. The groups are well matched on gender and parental education. Next. And the stimuli that we use are similar to before, just what I showed you. Next. But we also had additional stimuli, what we call wire stimuli, which are very similar. I can go into why we use that, and but for the time being, we're going to average across these two types of stimuli. Again, there's more to tell here, but I'm just kind of focusing on main results. Next. So here are the results. So threshold difference, fragmented minus illusory. So if you're doing better on the fragmented minus the illusory, if you're doing better on the illusory minus the fragmented, then you should have larger values here. So the larger values means you're doing very well on the illusory condition as compared to the fragmented condition. So as you can see, you get a nice group difference. Next. Next. And what was interesting is that these group effects were similar between first episode patients and later episode patients. So you get this group difference that emerges early in the illness and remains static thereafter. Next. Next. So completion deficits arise by the first psychotic episode and remain stable thereafter. Next. Some people say, oh, maybe this could be attributed to broad orientation tuning. Maybe patients have just trouble seeing the edges of the Pac-Man. Next. So what we did is we reanalyzed the data and removed patients who had any trouble on the orientation left-right condition. Next. And what we found is pretty much the exact same thing, and the effect sizes got larger. Next. Next. And the Cohen's D value increased from 0.64 to 0.84. So these are large effect sizes once you control for orientation, which was great. Next. We also found that the shape completion, the threshold difference, was related to cognitive disorganization. So the more disorganized they were, the more trouble they had in the illusory condition as compared to the fragmented condition. So again, this isn't just a generalized attentional problem or a problem of motivation, because it has to do with a threshold difference. So the more the threshold difference, the better the shape completion and the less disorganized symptoms they have. And this pattern was specific to disorganization. We did not find it with negative symptoms. We did not find it with positive symptoms. Next. Next. And we also, go back. We also showed that when we combined, this is one study, but when we combine it with a previous study with 110 patients, the effect was almost exactly the same, and it was highly significant. Next. We also showed that this effect is related to pre-morbid functioning. So the worse that they were doing before the illness onset, the more shape completion problems that they had. So it has to do with this specific type of patient. Next. So to summarize, shape completion deficits are medium to large in magnitude in schizophrenia. You get intermediate deficits in bipolar disorder. These deficits emerge fully by the first psychotic episode. They cannot be explained by poor attention or motivation because they're doing perfectly fine on the fragmented condition. Medication effects are unlikely since it's specific to one condition and since they didn't correlate with chlorpromazine equivalents in our prior studies, previous. And they're linked to cognitive disorganization and poor pre-morbid functioning. These deficits emerge relatively quickly. You can detect these deficits within 10 minutes of testing. And they're found with multiple methods across multiple studies using method of constant stimuli or staircase procedures. Next. Question is, okay, well, where do we go from here? There are different ways you can use these tests. One way is to predict a conversion to psychosis among people who are at clinical high risk, who have subthreshold psychotic symptoms, but who won't necessarily convert to a psychotic disorder. And so you can look for abnormal tests at baseline to see if that can help you predict better who will convert to a full-fledged psychotic disorder. Next. It can help you stratify patients into meaningful subgroups. So patients are very heterogeneous, as we all know. There are many different manifestations of the illness, and it probably is not one illness, it's probably several different illnesses. And so we could use these sorts of tests, both fMRI tests and behavioral tests, to kind of build these, to cluster patients into meaningful subgroups. Next. Also predict treatment response, which is completely nobody's looked at that. Next. And also could shed light on the pathophysiology, what aspects of the brain are changing when they get the illness, why are they changing, what are the behavior manifestations of this. Next. And it also can help us understand difficulties in vision, which themselves can be distressing or potentially debilitating, which is underappreciated aspect of the illness. Next. I would like to acknowledge members of my lab and also collaborators shown below at University of Rochester and also Rutgers University. Next. And also numerous other people who helped from Rutgers University, at Princeton University, and including Bart Kreckelberg and Michael Cole, Ravi Mill. You can obviously see them, but all of them played a role here in this research. Next. And I would like to say thank you very much for your attention for presenting to your group. And thank you for dealing with the technical issues that we encountered. And I welcome any questions. Next.
Obrigado. Thank you very much, Brian. Very interesting. I don't know about the people here, but for me, it was very, very interesting and enlightening.
Thanks so much.
I have a question. Did you have an opportunity to test whether antipsychotics help to improve? Because it seems to me biochemical imbalance initially that can be fixed with small quantities of antipsychotics and interrupt the process of getting worse. What do you think of that?
Yeah, it's a good question. It's something we always think about. So, I focused on two types of biomarkers. So let me focus first on the resting state. So the resting state biomarker actually, when you look at unmedicated or never medicated patients, they actually show stronger biomarkers, which means that it's not statistically significant, but we also have a small sample, but at least it's in the right direction. So to me, that indicates that if they are on antipsychotics, it's making their brain connectivity more similar to healthy controls. So obviously we need larger samples. But there have been other studies that have suggested similar things with thalamocortical connectivity. So I think that that's a reasonable thing to think. For shape completion, we did not find any relationships with antipsychotics. So, it's very imperfect. Correlating these things with chlorpromazine equivalents or olanzapine equivalents, it's very noisy, and the standardization methods are noisy. So I don't think anybody can say with certainty. But from what I've seen, it doesn't seem that it's really related to whether or not they're on antipsychotics.
Okay, I would like a comment. As you know, I have psychosis. I have schizophrenia. So what seems to me in an episode is that the thalamus doesn't filter. It passes, everything passes through. So it's very interesting the point you show, because sensory stimulus get through and you cannot filter it, okay?
Yeah.
But medication improves and filtering improves. So that's why I'm here.
I actually 100% agree with you that's a reasonable hypothesis. Because the thalamus, first of all, the thalamus is so complicated because we talk about it as if it's one thing, but there's actually a bunch of different functional organs that are all together in the same spot. And so we have some work that we're doing that we're trying to find the parts of the thalamus that are really driving this biomarker. So it's just preliminary data, but I definitely think that, if you think about what the thalamus is doing, it is a gatekeeper, and it's regulating activity cross cortex. So, and there's also phenomenological reports of sensory flooding, for example, that kind of are consistent with this idea that the thalamus is hyperconnecting and it's failing to prevent irrelevant information to be blocked. So I think that is a reasonable hypothesis.
And the antipsychotics are dead on. They help very much.
Yeah, it's a good point.
Okay, thank you very much. I will be passing a brief translation from what you said.
Yeah.
And I would like to thank you so much and hope you can come sometime. Okay.
Yeah, that would be fantastic. Next time around.
Yes. Thank you very much.
Thank you.
Bye.
Bye-bye.